Welcome to the Faculty Focus podcast, live from our annual Faculty Research Day with your hosts
Marleen
and
Teun,
highlighting the research of our professors and PhD researchers. Hello again. Next on the podcast we have Laura Mariën. She will talk about improving diagnostic accuracy and follow-up of neuroendocrine neoplasms using two novel technological platforms.
hi Laura, welcome. Can you briefly introduce yourself to our listeners?
Yeah, so I'm Laura Mariën. I'm a PhD student at the Center for Medical Genetics in Antwerp. I'm part of the Metgen research group and I'm focusing on oncogenetics of neuroendocrine neoplasms.
And what did you study before?
I've studied biomedical sciences here at the University of Antwerp.
Perfect, yeah. Well, we'll start with a broad question. Can you explain your daily work and research to a curious teenager who just started to love science?
Yeah, so I'm studying the oncogenetics of neuroendocrine neoplasms. These are quite a rare group of tumors that are also a really heterogeneous group of tumors. And these are quite challenging to diagnose and to follow up. So in my project, I want to discover the net-specific molecular alterations and to detect these alterations in the cell-free DNA of patients, to actually follow them up over time and to diagnose them.
So you want to find something specific that you can tie to those type of tumors.
Exactly. So we are looking at the methylation profiles that alter in these patients and use these as biomarkers to find them.
And the methylation, can you tell a bit more about the methylome that you research?
DNA methylation is actually an epigenetic modification in which a methyl group is placed on top of the DNA sequence at specific sites, at the cytosines. And by placing these methyl groups on top of the DNA, it becomes actually less readable for transcription. So these genes are not transcribed, for example. But in cancer, these methylation profiles change, during which then certain genes, like the oncogenes, become more prominent, and then the tumor suppressor genes are actually suppressed, which is something that we do not want to happen. And this is a process that happens in cancer cells, and which is a specific biomarker we can use.
Yeah, to research it. So those neuroendocrine neoplasms are different from other tumors. Can you explain a bit why?
Yeah, so the special thing about it is that it's originating from the neuroendocrine cells. I think one of the most well-known are the alpha and beta cells from the pancreas, for example.
Okay. Yeah.
So the insulinoma is, I think, one of the most well-known. But it's actually a whole spectrum of tumors that can originate from these neuroendocrine cells, and they can occur throughout the body, also in the lungs, for example, or in the gastrointestinal system.
So difficult to find as well.
Exactly, yeah. Because of their heterogeneity. It's not only clinical, some patients have very vague symptoms, but also molecularly there are a lot of differences. Also pathological-wise, it's not always that easy to discriminate them.
So it's mostly diagnosed late.
Yeah, exactly. So several patient organizations have already done research about this. And on average, a patient takes 5 years before he or she gets the right diagnosis.
Okay, so these type of tumors are not well known, or with the physicians neither, maybe.
Exactly. So you have very well-known experts in the field who know a lot about it, but the general practitioner, the physicians, do not always know about this type of tumors. And that's why they are called also the zebras of cancer.
The zebras.
Such
a nice animal.
yeah, for sure. But in medicine, during their study, they actually teach that when you hear the sound of hoofs, think horses, not zebras. So always think about what is most commonly happening. But neuroendocrine tumors are therefore called the zebras because they will always think about what's very common.
Yeah, but they also have hoofs.
Yeah, exactly. So they make the same sound.
Yeah, true. Okay, a nice analogy. So you're looking for new ways to diagnose as well. It's a bit an out-of-topic question, but compared to 20 years ago, there is more focus on patient well-being. Would you say that focusing on mental health is also a reason to rethink current diagnostics and follow-up tools?
Yeah, for sure. Especially follow-up. These patients need a lot of follow-ups, of course. And it's always quite stressful, I imagine, to have these scans and to wait a couple of weeks before you get the results. And biopsies, I think it's very invasive for the patient. So that's why we're focusing on taking a blood sample, which is, I think, somewhat less invasive.
Unless you're my husband and you fall down.
Yeah, but then we're also looking into urine and stuff for several cancers.
That will be easier.
Yeah, that will be easier. So, but that's not always possible for all types of cancers. But normally a blood sample, it's quite in the basic practice. In basic practice. So that's why we're focusing on this, to be able to diagnose and follow up patients.
I'm sure it will help a lot of people in the future. I'm very sure. Can you describe your dream day in the lab? What would make it absolutely perfect from start to finish?
That we have all really nice results, that our biomarkers can perfectly distinguish the tumors from the healthy controls, from other tumor types, and that we have the perfect model that we can bring to the patients. So,
is there a way to increase awareness for these type of tumors?
Yeah, we have a lot of very active patient committees, like, for example, INCA, that's worldwide. And we also have the 10th of November, which is the International Day of Neuroendocrine Neoplasms. So that's ways we try to raise awareness for this type of tumors.
Is there some advice you would like to give to new starting PhD researchers?
To not always think that everything will go perfectly the way you intended it to be, that there is a lot of administration as well on the side, which I didn't think of from the start, but it's there. But you learn a lot, and it's not only doing the science, it's also a lot of other skills that you will learn along the way. So it's just a great experience to do, actually.
And maybe also here think about the zebras and not only the horses.
For sure, but that's more like towards the general practitioners and like everyone, just think
keep
also.
Keep an open mind.
Yeah, keep an open mind and think about the zebras.
All right, well, thank you, Laura.
Which year of your PhD are you in now?
I just started my 4th year.
Okay, so one more to go.
Yeah, actually I have 2 more to go because I had 1 year as a PhD student, and during this year I applied for the FWO and I got the FWO, so I have another 4 years. Yeah, thanks.
Great.
Yeah.
So good luck.
Thanks.
And thank you for coming by.
Yeah,
no
thank
worries,
you for being here. Yeah, all right.
Thank you.